Good morning, everyone. Today, we will learn about beta oxidation, a crucial metabolic process in our body's lipid metabolism.
Fatty acids. A fatty acid contains a long hydrocarbon chain and a terminal carboxylate group. The hydrocarbon chain may be saturated with no double bond or may be unsaturated.
Functions of fatty acids. Fatty acids have four major physiological roles. 1. Fatty acids are building blocks of phospholipids and glycolipids. 2. Many proteins are modified by the covalent attachment of fatty acids, which target them to membrane locations. 3. Fatty acids are fuel molecules. They are stored as triacylglycerols. Fatty acids mobilized from triacylglycerols are oxidized to meet the energy needs of a cell or organism. 4. Fatty acid derivatives serve as hormones and intracellular messengers, e.g. steroids, sex hormones, and prostaglandins. Types of fatty acid oxidation. Fatty acids can be oxidized by 1. Beta oxidation. Major mechanism occurs in the mitochondria matrix. 2 C units are released as acetyl-CoA per cycle. 2. Alpha oxidation predominantly takes place in brain and liver. 1 carbon is lost in the form of CO2 per cycle. 3. Omega oxidation. Minor mechanism, but becomes important in conditions of impaired beta oxidation. 4. Peroxisomal oxidation, mainly for the trimming of very long-chain fatty acids. B-oxidation of fatty acid, the breakdown of a fatty acid to acetyl-CoA. Occurs in the mitochondria. Process is strictly aerobic. After production, acetyl-CoA is fed directly into the Krebs cycle. The beta oxidation of fatty acids involve 3 stages. 1. Activation of fatty acids in the cytosol. 2. Transport of activated fatty acids into mitochondria, carnitine shuttle. 3. Beta oxidation proper in the mitochondrial matrix. 1. Activation of Fa. This proceeds by Fa thiokinase, acyl-CoA synthetase. Present in cytosol. Thiokinase requires ATP, CoA-SH, Mg++. The product of this reaction is Fa acyl-CoA and water. Cytosol. Fatty acid plus CoA plus ATP. Fatty acid CoA plus AMP plus 2P. 2. Transport of fatty acyl-CoA from cytosol into mitochondria. Long-chain acyl-CoA traverses the inner mitochondria membrane with a special transport mechanism called carnitine shuttle.
Though transport of acyl-CoA into the mitochondria, rate-limiting step A. Acyl groups from acyl-CoA is transferred to carnitine to form acyl-carnitine catalyzed by carnitine acyltransferase I in the outer mitochondrial membrane. Though acyl-carnitine is then shuttled across the inner mitochondrial membrane by a translocase enzyme. The acyl group is transferred back to CoA in matrix by carnitine acyltransferase II. Finally, carnitine is returned to the cytosolic side by translocase in exchange for an incoming acyl-carnitine.
3. Proper of Acetyd. Oxidation in the mitochondrial matrix. There are 4 steps in oxidation. Step 1. Oxidation by FAD-linked dehydrogenase. Step 2. Hydration by hydratase. Step 3. Oxidation by NAD-linked dehydrogenase. Step 4. Thiolytic cleavage. Thiolase, alpha, C16. ARTH2H2H2CSCOA, palmitoyl-CoA. The first reaction is the oxidation of acyl-CoA by an acyl-CoA dehydrogenase to give a unsaturated acyl-CoA, enoyl-CoA. FAD is the hydrogen acceptor. The second reaction is the hydration of the double bond to hydroxyacyl-CoA, P, hydroxyactyl-CoA. The third reaction is the oxidation of B-hydroxyacyl-CoA to produce B-ketoacyl-CoA, an NAD-dependent reaction. The fourth reaction is cleavage of the 2-carbon fragment by splitting the bond between A and Acet carbons by thiolase enzyme.
Let's go through the 4 recurring steps of 1-beta oxidation cycle. In first step, palmitoyl-CoA gives trans-enol-CoA with the help of acyl-CoA dehydrogenase and produce FADH2. In second step, trans-enol-CoA by the hydratase hydroxyacyl-CoA. In third step, hydroxycyl CoA gives beta-ketoacyl CoA in the presence of beta-hydroxycyl CoA. Dehydrogenase by the formation of NADH+H+. In fourth step, ketoacyl CoA gives acyl CoA with the help of
the release of acetyl-CoA leaves an acyl-CoA molecule shortened by 2 carbons. This acyl-CoA molecule is the substrate for the next round of oxidation, starting with acyl-CoA dehydrogenase. Repetition continues until all the carbons of the original fatty acyl-CoA are converted to acetyl-CoA. In the last round, a 4-carbon acyl-CoA, butyryl-CoA, is cleaved to 2 acetyl-CoA.
Energetics of far oxidation, e.g. palmitic, 16 carbon. 1. Beta oxidation of palmitic acid will be repeated 7 cycles, producing 8 molecules of acetyl-CoA. 2. In each cycle, FADH2 and NAD plus H plus is produced and will be transported to the respiratory chain. FADH2 gives 2 ATP, NAD plus H plus gives 3 ATP, so 7 cycles gives total ATP, that is 5 into 7 is equal to 35 ATP.
3. Each acetyl-CoA which is oxidized in citric cycle gives 12 ATP. 8 times 12 is equal to 96 ATP. 4. 2 ATP are utilized in the activation of fatty acid. It occurs once. Energy gain equals energy produced minus energy utilized. Equals 35 ATP plus 96 ATP minus 2 ATP gives total 129 ATP. Conclusion. Beta oxidation is essential for energy production, especially during fasting or exercise. Produces large amounts of ATP by breaking down fatty acids to acetyl-CoA. Disorders can be life-threatening due to impaired energy supply. Brain tip for remember. 4 steps: oxidation, hydration, oxidation,